So today I want to talk to you about the scientific study of environmental toxins. We're going to be looking at how one would actually go about looking at and demonstrating the effects of an environmental toxin. And one of the words I'll be using is toxicant. And a toxicant is a toxin that is introduced into the environment by human beings or human activity. So you'll hear that word throughout this presentation, but I don't define it in the presentation. And the other thing we're going to look at is sort of the dose-response curve, which is the classic way of looking at the effects of toxins. There are two basic approaches to studying environmental disease. One is what's called the toxicological approach, and the other is an epidemiological approach. So let's look at each one of those in turn. Okay, first, toxicology. Toxicology is the study of the effect of poisons and toxicants on the animal body, and it's done in a lab setting. And the most common methodology to determine the effect of a given substance is to expose test organisms to precise doses in a laboratory setting, very highly controlled setting. The animals are then examined after a certain period for the occurrence of disease or other effects, and a dose-response curve is made, and we'll be looking at the dose-response curve a little later, but basically you look at the percentage of individuals that exhibit a given response at each dosage, and you plot that, and you look at what kind of response line, a function you get. High doses are often used in these studies in order to find the potential effects, in order to make sure effects actually occur. Use low doses and you're more likely not to see an effect. So the point in these studies is to really see effects of these toxins. And then you can go back and do studies at lower levels later on once you've established that the toxin is a problem. And the effects on humans often must be extrapolated from the curve because the lab exposure dose is often high, and humans are much more massive than lab test animals like mice and rats. So that's a toxicological approach. An epidemiological approach instead looks at populations of people who have been exposed to a toxicant and compares it with similar populations of people who have not been exposed to the toxicant. So you look at historical exposure, usually of a group of humans, so it's exposure that occurred in the past, and you see if that exposure has increased the rate of disease or other health effects in comparison to unexposed control groups. And finding a control group that is similar in all other aspects to the exposed group is really a difficult task, and it's what you learn when you go to grad school and try to become an epidemiologist. Each approach has its particular advantages. For example, toxicology, you get the data relatively quickly, you control a single variable, the dose of the dose that the animals are exposed to is known precisely, and the effect, if present, is likely to be observed. In contrast, epidemiology has the advantage of using human subjects. You have a genetically diverse group that is examined. You have exposure levels that are realistic because that's what really happened to these humans. You get to examine longer term effects, and you have a very large sample size. So those are the advantages of epidemiological approaches. Now, one of the things you'd be asked for on exams like the AP is, can you think of the disadvantages of each? And I wanted to say the disadvantages of each are taken by negating the advantages of the others. So, for example, the disadvantage of epidemiology is that it takes a while to get the data, the opposite of a toxicology study. There is not easy control of a single variable. There are lots of variables that are changing in that population. The dose is not known precisely. Each person was exposed to a different amount. The effect, if present, is much less likely to be observed. And those are the disadvantages of an epidemiological approach. And I'll leave you to tell me the disadvantages of a toxicological approach. But if you said, oh, you don't use human subjects as the first one, you're on the right track. In the best case, both types of evidence are used. You have, even though that means some human beings have been exposed, but given that human beings were exposed, which is usually when we're doing these things as a rear guard action, it's nice to have both types of data in hand. Now let's look at dose response curves, and here are some examples of them. Dose response curves are generated by plotting dose on the x-axis and response on the y-axis. In the graphs below, the dotted line is the line of no effect. Points below the dotted line indicate a beneficial effect, and those above it a detrimental or a harmful effect. The curves listed below show three kinds of dose responses. The first one is any dosage causes an effect, and that is linear related, and that effect is linearly related to the dosage. In the second one, low dosages cause no response until you reach a threshold level, at which point you then get a linear response. And the hormetic model, which I'll talk about in a little bit, shows you that at low doses you actually get a beneficial effect, and then at some threshold dose it turns around and starts going up, and at a second threshold you actually finally get a negative effect. And we'll talk about hormesis a little later. Now, one of the things that you measure in terms of toxicity, especially lethal toxicity of a substance, is what's called the LD50. And the LD50 is the lethal dose to 50% of the test organisms. So you expose a bunch of test organisms to varying amounts of a chemical, and the next thing you know, some number of them die, and you plot that, and then you calculate the dose at which 50% would be predicted to die, and that's the LD50. It is mandatorily determined for all new synthetic chemicals, so somebody's got to be doing that. It is measured in interesting units, milligrams of toxin, or mass of toxin really, usually milligrams, per kilogram of body mass. And you can see in the chart at right that aspirin has a lethal dose of 1,750 milligrams, or 1.75 grams per kilogram of person. And since most of you are between 50 and 75 kilograms, you could figure out what a lethal dose would be. One thing that may not be immediately obvious, but you should make sure you understand, is the smaller the LD50, the more lethal the chemical, right? So sodium cyanide has a small LD50, 10 milligrams per kilogram. A 100 kilogram person can only be exposed to 1,000 milligrams, or 1 gram of cyanide, and that's deadly. Now, you can take this LD50 approach to look at any health effect, and when you do that, you call it the effective dose. And the effective dose 50 is the dose at which 50% of the population exhibits whatever non-lethal effect is under study. For example, skin rashes, or lung disease, or high blood pressure, or whatever you're looking for, or immune system suppression. So the effective dose is often very important for the kinds of things we're looking at in this course, where people aren't dying directly from the toxin but are suffering long-term health effects. Now, this toxicity itself is highly complex, and these two curves are meant to represent some of that. First of all, many toxins behave in a non-linear fashion and even more complex ways. And you can see the curves in this one are linear over short portions. For example, this first curve is linear from approximately here to about here, but non-linear at the endpoints. So many toxins have even more non-linear curves. Second, note that we use a logarithmic scale when we measure this. In other words, each line on the horizontal axis, represents a 10-fold increase in the dose. So that's an important thing to note, that we are often measuring these in 10-fold increases in our experiments. Finally, notice in the second graph that the graph for A shows a lower ED50, which means it is more toxic at the ED50 level. But the B graph shows a larger effect for low doses. So it just shows you that the ED50 or the LD50 may not tell you the entire story of a toxicant. So As I already said, the LD50 and ED50 may not indicate the relative toxicity for all doses. We saw that in the last one. There may be nonlinear effects. Most studies are done at high dosages and then calculated backwards using a linear model. May not always be correct. Low doses may have a long tail. In other words, there may be effect at low doses for very low doses. And in the extreme, there may not be a safe dose at all, right? There may be no safe level, as we looked at earlier. So those are some complications of the dose-response curve. Finally, I want to talk about hormesis. Hormesis is a term used by toxicologists to refer to an environmental agent that has a beneficial effect at low dose and a negative effect at high dose. So what does that mean? Well, it's kind of similar to that old adage, what doesn't kill you makes you stronger. This was depicted in the third graph that we saw earlier, where there was a beneficial effect at low dose. An example might be the often cited studies about alcohol consumption, where people who have 1 to about 2 drinks a day seem to have certain health effects that are better than people who don't drink. That's controversial, by the way, as a result, but many people stand by those scientific studies that show it. And hormesis in general is a controversial hypothesis. So that's it for now. I hope that today you understand the difference between epidemiology and toxicology, and I hope you understand how a dose-response curve would be calculated. In the next video, we're going to see all the different models of toxins, starting with the classic model of a toxin, and many people, that's the only model of a toxin that they are willing to admit. But as you will see, there are many other models for toxins that are really occurring in the real world and mean that the classic model can't be used. So we'll see that next time. And until then, you know what you should be doing. Study hard.